Gene therapy. This is the point in the course where we move away from discussing technology in abstract terms and begin examining technologies that intervene directly at the level of human biology. Specifically, we are now dealing with systems that can modify the genetic instructions encoded within your cells. That distinction is critical because most technologies we have discussed so far operate externally, influencing behavior, environment, or systems. Gene therapy operates internally at the molecular level, where biological function is defined. This session will focus on understanding how gene therapy works, what it enables, and what it demands from society in terms of regulation, ethics, and governance. There are over 7,000 identified genetic diseases worldwide. At present, only around 20 gene therapy products have received regulatory approval. However, the market for gene therapy is projected to grow by roughly 10 times its current value by the year 2030. These figures indicate that the field has already transitioned from experimental research into clinical application and commercial expansion. Gene therapy is no longer a future concept. It is an active and rapidly scaling sector of modern medicine. The Philippines is directly affected by these developments. More than 1 million Filipinos carry hereditary conditions that are already being targeted by therapies under development. This means the relevance of gene therapy is not hypothetical at the national level. The implications are immediate, and the decisions surrounding access, policy, and infrastructure will shape how these technologies impact Filipino populations. Traditional medicine typically addresses diseases after they manifest, either by alleviating symptoms or by modifying biological pathways associated with disease progression. Gene therapy represents a fundamental shift from this model. Instead of acting on downstream effects, it targets the source of the problem by modifying or correcting the DNA sequence responsible for the condition. This approach reframes treatment as an intervention at the level of biological instruction. Within a science, technology, and society framework, this raises important implications. When DNA is modified, the change is not simply therapeutic but structural. In somatic cells, these changes affect only the treated individual. However, if modifications occur in germline cells, such as sperm, eggs, or embryos, those changes can be inherited by future generations. This introduces a level of consequence that extends beyond individual patients and into population-level and intergenerational considerations. Gene therapy can be categorized into 3 main approaches. The first is gene addition, where a functional copy of a gene is introduced to compensate for a defective one. The second is gene silencing, which involves suppressing or disabling genes that produce harmful effects. The third is gene editing, which directly alters the DNA sequence at a specific location. Among these, gene editing represents the most advanced and controversial approach. Technologies such as CRISPR enable precise modifications, but they do not inherently distinguish between therapeutic correction and enhancement. The same mechanism used to fix a disease-causing mutation can also be used to alter traits that are not medically necessary to change. As a result, the boundary between treatment and enhancement is not determined by the technology itself, but by societal norms, regulatory frameworks, and ethical considerations. A major technical challenge in gene therapy is not only designing the appropriate genetic sequence, but also delivering it effectively into target cells. Viral vectors are currently the most established method, utilizing modified viruses to transport genetic material into cells. Lipid nanoparticles represent a newer and increasingly important platform, particularly because of their scalability and successful use in mRNA-based COVID-19 vaccines. These delivery systems are critical because even well-designed therapies can fail if the genetic material does not reach the intended cells in sufficient quantity or triggers adverse immune responses. In practice, delivery limitations remain one of the primary reasons why gene therapy trials do not advance successfully through clinical stages. CRISPR-Cas9 originated as a defense mechanism in bacteria, allowing them to identify and cut viral DNA. In 2012, researchers adapted this system into a programmable tool for genome editing. By designing a guide RNA, scientists can direct the Cas9 enzyme to a specific DNA sequence where it introduces a cut. The cell's natural repair processes then enable targeted modifications, such as insertion, deletion, or correction of genetic material. This innovation has significantly accelerated the development of gene editing applications. Within a decade, it led to the first approved CRISPR-based therapy, known as Casgevy, which demonstrated functional cure rates exceeding 90% in certain clinical trials. A critical implication of this technology is its accessibility. CRISPR techniques are now teachable at the undergraduate level, which means the capability to edit genetic material is no longer confined to highly specialized institutions. This widespread accessibility introduces new challenges in governance, oversight, and responsible use. The development of gene therapy has been shaped by both progress and setbacks. A defining moment occurred in 1999 with the death of Jesse Gelsinger, a participant in a gene therapy trial who experienced a severe immune reaction to the treatment. This incident led to a significant slowdown in research and prompted a reevaluation of ethical standards, informed consent processes, and regulatory oversight. The resurgence of gene therapy in the following years was accompanied by stricter safety protocols and improved clinical frameworks. This history illustrates that advancements in the field are not solely driven by scientific breakthroughs, but also by the lessons learned from past failures and the resulting improvements in governance. In the Philippine context, hereditary diseases present a measurable and significant burden. Glucose 6-phosphate dehydrogenase deficiency affects an estimated 3 to 9% of the population. Beta thalassemia is another condition with serious health implications. Notably, there are emerging gene therapies that offer functional cures for some of these diseases. However, the cost of such treatments can range from $2 to $3 million per patient, making them inaccessible to the vast majority of Filipinos. Additionally, these treatments are not covered by national health insurance systems, such as PhilHealth. This creates a situation where the scientific capability to cure exists, but access is limited by economic and systemic barriers rather than by technological constraints. The Philippines has several policies related to biotechnology, biosafety, and ethics. However, as of 2024, no gene therapy product has been formally registered with the Food and Drug Administration of the Philippines. Furthermore, regulatory pathways for advanced therapy medicinal products, which include gene therapies, are still under development. This indicates a gap between global scientific advancement and national regulatory readiness. Even if patients have the financial means to access treatment, the domestic infrastructure may not yet support its safe and legal implementation. Bridging this gap requires coordinated efforts in policy development, regulatory capacity building, and healthcare system preparedness. Gene therapy has already demonstrated significant clinical successes. Conditions such as sickle cell disease, which previously required lifelong management, now have documented cases of functional cure. CAR T-cell therapies have achieved high remission rates in certain types of leukemia. The mRNA platform, proven effective during the COVID-19 pandemic, is being adapted for broader therapeutic applications. These examples confirm that the scientific foundation of gene therapy is robust. The primary challenges now lie in scaling these solutions, ensuring equitable access, and integrating them into existing healthcare systems. Despite its promise, gene therapy presents several risks and ethical challenges. Scientific concerns include off-target effects, where unintended genetic modifications occur, immune responses to delivery mechanisms, and uncertainty regarding the long-term durability of treatments. Beyond these technical issues, there are broader societal concerns. High treatment costs create disparities in access, raising questions about equity. The case of He Jiankui, who conducted unauthorized gene editing on human embryos, highlights the risks associated with inadequate oversight. Another critical issue is the distinction between therapeutic use and enhancement. Since the technology itself does not differentiate between these purposes, it is up to regulatory bodies and societies to establish and enforce boundaries. These challenges emphasize that governance and ethical considerations are integral to the responsible development of gene therapy. ongoing research continues to refine gene editing technologies. Base editing and prime editing offer more precise methods with reduced risk of unintended DNA damage. Advances in computational tools, such as AlphaFold, have significantly improved the ability to predict protein structures, accelerating the identification of therapeutic targets. While these innovations expand the potential of gene therapy, they also increase the urgency of developing appropriate governance frameworks. The pace of scientific advancement raises questions about whether regulatory systems can adapt quickly enough to manage emerging risks and opportunities. At the international level, there is a growing consensus that clinical applications of heritable genome editing should not proceed at this time due to unresolved ethical and safety concerns. However, such consensus statements do not carry legal authority. In the Philippines, existing laws provide a foundation for ethical oversight, but gene therapies are not yet fully incorporated into regulatory frameworks. The most immediate governance challenge is ensuring that patients with treatable genetic conditions can access care within the country. Addressing this issue requires not only policy development, but also investment in healthcare infrastructure, regulatory capacity, and public engagement. The next part of the session involves evaluating 3 scenarios related to gene therapy applications. For each scenario, consider whether the application is acceptable and determine who should have the authority to make that decision. Reflect on the basis for these decisions, including ethical principles, regulatory frameworks, and societal values. This exercise is designed to highlight the complexity of decision-making in the context of emerging biotechnologies. Gene therapy should be understood as part of a broader technological and societal landscape. It intersects with developments in climate-related health, nanotechnology, and digital information systems that influence public understanding and perception. The central issue is not solely technological capability, but the structures through which decisions are made. These include political processes, ethical frameworks, and democratic participation. The development and application of gene therapy ultimately depend on how societies choose to govern and integrate these technologies. The key question is not whether gene therapy is possible, but how it will be managed, who will benefit, and what values will guide its use.